Guys, tonight's little podcast is going to be about types of crosses, so looking at monohybrid and dihybrid crosses, what ratio means, and also looking at linked genes. So first of all, we should go through the types of inheritance. The first type of inheritance that we learnt was complete dominance. So that is where one allele is dominant over the other, so that the phenotype of the heterozygote, so in this case the example I've given is tongue rolling, you could have capital T, little t, is the same as the homozygous dominant, which in this case would be capital T, capital T. So you can see in this case that both of those, even though their genotype's different, would have the same phenotype. So that is complete dominance. You're either recessive or you're dominant. The second type of dominance that we've learned about is codominance. Now in this case, you have, well, the example that we always use is blood type, where you have 3 alleles present on the one gene. So in this case, it's where this heterozygote exhibits the phenotype of both homozygotes. So with blood type, you have A, B, and O alleles. A and B are both what we say codominant over O. So that means if you had the alleles A and B in your blood, you would have type AB blood. Now, the other thing that we've learned is the different types of crosses. So a monohybrid cross, as you would have learned, is a cross between, initially, 2 true breedings, so that means homozygous parents that differ in a single trait. That's where mono comes from, for its single trait here. The subsequent generation, which is called F1, so this is after you've crossed the 2 parents, which are called P, are all heterozygous for that one trait. So again, going with that tongue rolling example, F1 would all be big T, little t. The other type of cross that we're looking at today is dihybrid crosses, and this is a cross again between 2 breeding parents that differ in 2 traits. So the example I've given here is the P's example, where big S stands for smooth, little s stands for wrinkled, big Y stands for yellow, and little y stands for green. So we're looking at 2 separate genes here for different traits. Again, when you cross the 2 breeding parents, the subsequent generation, F1, will all be heterozygous for both traits, so they will all look like this. So let's have a look at some ratios that we get. Now, again, I've gone with the tongue rolling, and these individuals here are from F1, so the first generation after you've crossed the parents. All right, so both of them are heterozygous. When we cross those together, this is what we get. So we've got this genotype here, homozygous dominant. We've got 2 with the heterozygous dominant and 1 that's homozygous recessive. Now, when we talk about ratios, we're talking about the phenotype. So in this case, as I've marked in red here, we've got 3 here, which are tongue rollers, and 1, which is a non-tongue roller. Now, if you are asked to write about phenotypic ratio in your exam or SAC, you need to make sure that you've listed also the phenotype there, because that's a common mistake, is not to list that. And this obviously is an example of complete dominance, because you've either got a recessive trait or a dominant trait. This is another type of inheritance, so codominance, which we mentioned before. Now, as I said with codominance, 2 of these alleles are going to be both codominant, so both expressed. So if we look at what happens with this cross, A, so crossing A obviously gives you blood type A. Crossing an A and a B gives you AB type blood. And again, this is an AB type blood. And then these 2 are going to give blood type B. So we get a different ratio this time. It's a bit different to the 3-to-1 that we saw before, so it can't be complete dominance anymore. If we get, so this ratio is 1 to 2 to 1. So if you see that ratio appear, this is now codominance. So 1 to 2 to 1. That tells you it's codominance. The other question I get about blood type is what this little, or capital I guess, means. And it just means, that's just showing you that it's looking at blood type, because as you would have remembered in Unit 3, we look at how blood type can clot, so these A's and B's represent the antigens that are present in your blood. And as you would remember, blood type O, which is not shown here, doesn't have these antigens, so it's actually missing. Now, test crosses is another thing that appears quite a bit. And when we're looking at a monohybrid cross, a test cross is used to determine whether an individual exhibiting a dominant phenotype is heterozygous or homozygous. Because remember, with the tongue rolling example, if you can roll your tongue, you could be heterozygous for that trait, so capital T, little t, or you could be homozygous dominant for that trait, so capital T, capital T. What a test cross enables you to do is actually determine if your genotype is heterozygous or homozygous. And the most important thing to remember with a test cross is that you're crossing it with a homozygous recessive phenotype. Now, the reason you're doing that is because obviously any individual exhibiting a recessive trait, you know with 100% accuracy what their genotype is. Now, looking at the results could mean different things. So this is an example of a test cross 1. So again, because it's a test cross, this is our recessive genotype that we're crossing it with. And in this case, we're crossing it first with a heterozygote. Now with these results, you'll notice that half can roll their tongue and half of the offspring can't roll their tongue. So if this appears in the pedigree here, sorry, the Punnett square, or in the offspring I should say, you'll notice that the individual that you didn't know must have been a heterozygote. Now with test cross number 2, we've crossed a homozygote dominant with a recessive, homozygous recessive one, again because it's a test cross. Now in this case, you'll notice that all offspring can actually roll their tongue and none of them can't roll their tongue. So if all offspring exhibit the dominant phenotype, then the unknown genotype must have been homozygous. Now this is the last type of cross that you will come across in this unit, and this is called a dihybrid cross because you're actually looking at 2 separate genes. So in this case, the S stands for, or capital S stands for smooth, little s stands for wrinkled, big Y is yellow, little y is green. I think I mentioned all that before. And what you've done in each box here is you've separated the different alleles to represent the gametes that could result. Okay, so the same as down here. So both of these originally would have been a big S, big Y, little y that you've crossed together. Now let's have a look at what happens when you cross all these together and what offspring result. So here you go. Now the ones in red are the ones that are going to show smooth and yellow phenotypes. The ones in blue are the ones that are going to show smooth green phenotypes. The ones in green, I probably should have done that the other way, are the ones that are going to be wrinkled and yellow. And then this one here is the one that's going to show both recessive traits, so wrinkled and green. So the ratio is now 9:3 to 3:1. And that ratio that you get is typical of a dihybrid cross. When we get that ratio though, we're making one huge assumption, and that assumption is that these 2 genes, so the one coding for colour and also shape of these peas, are independent of each other and they're independently assorted during meiosis. Now sometimes that doesn't exactly happen, and we've talked in class about when the genes are very closely located on a chromosome, so close together, often they can be what we call linked. So this law that we've been talking about, which we make that big assumption on, is called Mendel's law of independent assortment, and it means that genes for different traits assort independently of one another. That just basically means that if a pea is yellow, doesn't mean that the shape then has to be smooth. It could be a combination. So how do you actually check that genes for different traits are assorted independently? Well, the answer is to do another test cross. So with a dihybrid test cross, again, because you're looking at 2 genes, right? What you need to make sure of is the one that you're crossing it with is recessive homozygous for both traits. So this is an example here. So we've crossed this individual here, which is obviously heterozygous for both traits, with a recessive homozygous one here. Now, if they're independently assorted, this is the ratio we should get. So we should see roughly equal proportions of each of these four phenotypes here, which are yellow smooth, smooth green, wrinkled yellow, and wrinkled green, right? We should see equal proportions of those. So that if you see that presented as some sort of data in your SAC or exam, that's what it's telling you, that they're independent of each other, and most likely that these genes are located pretty far apart on a chromosome. If you're presented with some sort of data though that shows this, then this is indicating that linkage has occurred. So these red dots here are indicating what we call the recombinants. Now the recombinants are individuals with a genotype that are different to what the parents, the initial parents had. So you've got to think back. Parents are always true breeding, right? So the parents that would have been crossed here, one would have been purple long and the other one would have been red short. So they're what we call the parental phenotypes here, right? So we've got a much higher proportion of the parental phenotypes than what we call the recombinant phenotypes. So they're these ones in the middle. If you see that sort of data being represented, right, so you wouldn't be asked to do a cross and then determine if they're linked or not. They would give you some sort of data, and this clearly indicates that there's some sort of linkage going on. So both red short are linked, so those two genes, and purple long are linked. So if they weren't linked, this is the ratio we would expect here. I know it's in huge numbers, but what sort of ratio does that look like? You'll notice it's actually in the 9:3 to 3:1 ratio, and that's what we expect when we're looking at two genes, right? So a dihybrid cross if they're not linked, so independently assorted according to Mendel's theory. But we know, obviously now, with a lot of research, that that's not always the case. And this is what's happened with what we call linkage. So I hope that's made a little bit of sense today. Obviously, good luck with your SAC, and I hope there's lots of questions tomorrow in class. Thanks, guys. Bye.